Welcome to our first video on the A-level biology syllabus. Today we're going to be jumping into an introduction to biological molecules. Okay, so let's jump straight into the three fundamental types of chemical bonding: covalent, ionic, and hydrogen bonding. Each of these bonds plays a crucial role in the structure and function of biological molecules, which is why it is important to understand them in detail. Let's start with covalent bonding. Covalent bonds form when two atoms share a pair of electrons. This sharing allows each atom to achieve a full outer electron shell, which is generally a more stable configuration. Covalent bonds are particularly common in organic molecules, which are rich in elements like hydrogen, carbon, and oxygen. For instance, the bonds between the carbon and hydrogen atoms in methane are covalent. One important point to remember is that covalent bonds can be either single, double, or even triple bonds, depending on how many pairs of electrons are shared. A single bond shares one pair of electrons, a double bond, two pairs, and so on. The strength of a covalent bond increases with the number of shared electron pairs, so a double bond is stronger than a single bond. This bond type is essential for the structure of biomolecules like DNA, proteins, and carbohydrates. So next we have ionic bonding. Ionic bonds are formed through the transfer of electrons from one atom to another, creating ions. One atom loses one or more electrons to become a positively charged ion, while the other gains those electrons to become a negatively charged ion. These oppositely charged ions are then attracted to each other by electrostatic forces, creating an ionic bond. A classic example of an ionic bond is sodium chloride, or table salt, where sodium loses an electron to become Na+, and chlorine gains an electron to become Cl-. Ionic bonds are generally stronger than covalent bonds in the solid state, but they can be disrupted in solution. In biological systems, ionic bonds play important roles, especially in the function of electrolytes such as sodium, potassium, and calcium ions, which are crucial for nerve signaling and muscle contraction. Finally, let's discuss hydrogen bonding. Hydrogen bonds are weaker than covalent and ionic bonds, but they are still incredibly important in biology. Hydrogen bonds occur when a hydrogen atom, which is already covalently bonded to an electronegative atom, such as oxygen or nitrogen, is attracted to another electronegative atom nearby. For example, in a water molecule, each hydrogen atom is covalently bonded to an oxygen atom, but water molecules also form hydrogen bonds with each other. These bonds are responsible for many of water's unique properties, like its high boiling point and surface tension. Hydrogen bonds are also crucial in the structure of DNA, where they hold together the complementary base pairs. While each individual hydrogen bond is weak, together they create a strong and stable structure that can still be opened up during processes like DNA replication and transcription. So now let's talk a little bit about monomers and polymers, key concepts in biology, especially when studying macromolecules such as carbohydrates, proteins, and nucleic acids. Starting with monomers, these are small single molecules that can join together to form larger structures. Think of monomers like individual building blocks. Each monomer has certain functional groups that allow it to bond with other monomers in a specific way. When monomers join together, they create polymers, long repeating chains of these smaller units. A simple example of this is glucose, a monomer that joins with another glucose molecule to form starch or cellulose, which are polymers. The process that links monomers together to form polymers is called a condensation reaction, sometimes referred to as dehydration synthesis. Each time a new monomer is added to a growing polymer chain, a molecule of water, H2O, is released. This happens because parts of the water molecule, specifically a hydrogen from one monomer and a hydroxyl group, OH, from another, are removed to allow a new bond to form between the monomers. This process is key in biology. For instance, it's how amino acids link up to form proteins and how nucleotides join to form DNA and RNA. Condensation reactions are essential for building the complex macromolecules that make up living organisms. However, just as polymers can be built up, they can also be broken down, which is done through hydrolysis reactions. In hydrolysis, water molecules are added to break the bonds that hold monomers together in a polymer chain. When water is added, it splits into H and OH, which then attach to the separate monomers, effectively breaking the bond between them. This process is crucial for digestion. For example, when you eat a starch molecule, enzymes in your digestive system add water molecules through hydrolysis to break down the starch into glucose monomers, which can then be absorbed and used for energy. Let's quickly review these essential concepts. In terms of bonding, we have three key types: covalent bonds, where atoms share electrons; ionic bonds, where oppositely charged ions attract; and hydrogen bonds, which are weaker attractions between hydrogen and electronegative atoms. Then we have monomers, single units that link together to form polymers or long chains. This linking happens through a condensation reaction, where water is released as each monomer joins the chain. On the other hand, hydrolysis reactions break polymers back down into monomers by adding water. These reactions are fundamental to building and breaking down the molecules our bodies rely on. Understanding them is key to mastering biological chemistry. Thank you very much for taking the time to watch this video, an introduction to biological molecules. Make sure you like, comment and subscribe, and check out other videos on the A-level biology syllabus that we have on this channel.